obtained to date. All other.
We can hear you just fine.
Loud and clear.
Yep, you're good. Go ahead and get started.
Okay, cool. Okay, so hey guys, my name is Anirudh, and I'm a junior at Thomas Jefferson High School for Science and Technology, and today I'm going to be hosting the first workshop, the applications of deep learning techniques to computational biology problems. Okay, so just as a brief introduction, like some definitions that you guys are going to need to know is number one, deep learning, and number two, computational biology. So basically, deep learning is a form of machine learning in which like networks such as neural networks are trained in order to learn relationships in a pre-existing data set, so that like if there's a new data set that's presented, it would be possible to make accurate predictions about those new points. And then computational biology is a bit of a broad term, but it's basically the application of modeling techniques to study biological systems. Okay, so today I'm going to go over 4 different applications. So the first one is modeling the fitness of unregulated RAS mutants using computational mutagenesis and machine learning. Okay, so just for some background information, RAS proteins are basically proteins in the human body that are responsible for cell growth, and they exist in 2 possible states, on and off. So the middle diagram that you see on the screen is basically a ribbon diagram of the RAS protein. So basically in this research, it was important to just get the ribbon diagram, and I'll explain more like later how we use that. But yeah, so just for some background information, RAS proteins can exist in 2 states. Okay, so basically the key part of the research is looking at what happens when there's mutations to the RAS protein. So basically what happens is if the protein structure is mutated, sometimes it can be left in a permanently on state. And if it is, then tumor growth and even cancer development can result. So obviously this is pretty problematic. So it's pretty important to like research the protein structures and see like what precisely every single mutation has on the structure of the protein, and like whether it's possible to like predict ahead of time whether the resulting tumor would be malignant or benign. So the middle diagram is basically a Delaunay tessellation, which I'll also be talking about more in detail later in the presentation. But basically in the research, we used it to pinpoint like what the structural effects on the RAS protein structure would be if there's like any particular mutation. So like you can see at the bottom, there's like a red dot. So that was basically like, first of all, every dot in this structure represents an amino acid. So that red dot was like the site of a mutation, like of a single point amino acid mutation. So it could have been changed from like an A to a C, for example. And when that happens, basically the benefits of using a Delaunay tessellation is that you can kind of trace and see like what the cascading effect of that mutation is throughout the entire structure of the protein, instead of just like a localized effect. So you can kind of see like how it connects to different edges and vertices, and like how it ultimately affects the entire structure. But yeah, as some more background information, so 30% of all human cancer is actually linked to mutations to these RAS proteins. And it's not all mutations of RAS proteins that necessarily cause cancer. Like there are actually some mutations that could somewhat be beneficial and leave the protein in like a better structure than it was before the mutation. But when there are mutations to this protein, there can be some pretty bad consequences. Okay, so in particular, pancreatic cancer actually has a 95% chance of being caused by mutation to the RAS protein out of all the different proteins. So yeah, it's definitely pretty important to study this protein. Okay, so the basis of the research relied on a data set that was gathered from a recently published paper in the Nature journal. And they basically experimentally determined what the structural and functional changes of single point amino acid RAS mutations were. So they measured the functional changes through what's called fitness values, and there's specific formula you can see on the like bottom left. But basically, like they conducted it in a lab setting, and this actually had like a lot of problems because the technique they used was called experimental mutagenesis, and this has like a lot of downsides. Number one, it's pretty costly. Number two, it's inefficient, and because of those two factors, it's automatically not able to be scaled to like larger projects or larger research that needs to be conducted in the future. And then also another downside of like experimental mutagenesis in the lab was that it's bad for the environment because they used a lot of tools in the lab that have plastic, for example. So it was definitely important to find like an alternative way than like an experimental mutagenesis method. Yeah, so yeah, this slide basically just sums up what I said before. In total, there's 3,135 single point amino acid mutations to the RAS protein structure, and experimentally determining it is complicated, expensive, and time consuming, and also bad for the environment. So basically the focus of this research that we conducted was to develop a novel approach that utilized computational mutagenesis and machine learning to predict the same functional effects of RAS mutants, but by virtue of having everything done through computational mutagenesis and machine learning, we didn't actually have any of those downsides from the previous research because like literally everything was done through a computer. So that had a lot of benefits because we didn't have to spend a dime. It was extremely efficient. We could do it in like a couple weeks, as opposed to like the several months that the researchers took. And of course, we didn't use like experimental tools like that had plastic. So there are a lot of benefits to using this technique. So the computational mutagenesis was the first part of the research, and basically this technique allowed us to essentially quantify the 3D protein structure of a resulting RAS mutant. So like we could essentially, using a DNA tessellation, take the protein structure of a RAS, the protein structure of a RAS mutant and like assign an energy score, which essentially was a measure of like how badly affected that structure of the protein was. So it was a pretty complicated technique, but yeah, that was ultimately what the purpose of it was, to like provide a numerical score to each of those 3,135 RAS mutants. And then the machine learning was also equally as important because basically based on the central dogma of molecular biology, which states that the structure of a protein determines its corresponding function. Because of that property, we were able to, from this computational mutagenesis technique, then utilize machine learning models to predict the functional effects of those RAS mutants based on those like quantifications and energy scores. So just to reiterate, we had the data set from that one research team, and with the computational mutagenesis method, we were able to quantify the structure of each of those mutants. And then with the machine learning from the structure, we were able to predict the functionality of those mutants. So by the end of the research, we could essentially determine for every single one of those 3,000 mutants, like whether each one would be malignant or benign. Okay, so back to the computational mutagenesis technique. Here's just like a visualization of the process. So it was a 3-step process. So first in figure A, that's just the ribbon diagram of the RAS protein. And we were able to, for any of you guys interested in potentially replicating this, we were able to develop it on the Chimera software. And basically from figure A, that's what we needed to generate a Delaunay tessellation, which you see in figure B. So a Delaunay tessellation is essentially like a 3D tetrahedral mapping of a RAS protein structure. So you can see in the Delaunay tessellation that like a bunch of points and a bunch of like vertices connecting those points. So every single point represents an amino acid of the protein structure of RAS, and then the vertices are just essentially like they don't actually occur in real proteins, but it's just like for us so we could see like what effect one amino acid had on an adjacent amino acid. So that's why we drew those different vertices. And basically, from figure B, we then had to modify it to like a modified delineate tessellation, which you see in figure C. And that essentially just takes out all the vertices that were of length greater than 12 angstroms, because essentially we determined the like 2 amino acids that are connected and they're like greater than 12 angstroms away. They essentially have like a negligible effect on each other, to the point like it doesn't really affect the results of our research. So we just were able to take it out and also like simplify the process and made our like the efficiency of the models much quicker. So then from figure D isn't like actually a step, it's just a demonstration of how this delineate tessellation could be used in our research. So basically, if that red dot, which like I said before, represents an amino acid, was mutated from like an A to a C, then with the delineate tessellation structure, we essentially had like a systematic way of like tracing the cascading effects of that mutation on the rest of the protein structure. So we could see like every single connection that that amino acid had to other amino acids, and then those connected amino acids, their connections to other amino acids. So it just like it was like a spiraling process, and it kept going down throughout the entire protein structure. So and we were able to do this for every one of the 35 mutants of the RAS protein structure. Okay, so once we were able to generate the delineate tessellation for each of the mutants, the next step in the computational mutagenesis technique was to essentially quantify like the structural effects of every resulting mutant. So in other words, like we had to come up with an energy score for it. So to make the energy score, we relied on two metrics. One is called residual score, and the other is called a residual profile vector. So essentially, the residual score was it took like in a particular mutation of the structure, it looked at like a single amino acid that was mutated, and it essentially summed up the energy scores of all the quadruplets of amino acids surrounding it. So essentially what that means is that like if you see in the bottom right, that red dot, so that would, for example, be the site of the mutation for that particular mutant. And we would look at every pair of 4 amino acids. So like the red dot and then DAL, like the red dot and ALF, and like just all the pairs of all the quadruplets of amino acids. And with that, for each of those quadruplets that was connected to that central amino acid, we're able to assign an energy score to each one of those. And then ultimately the residual score was just for that particular amino acid in the structure, the sum of all those energy scores of each of those quadruplets. So that was the residual score. But then for the that's just looking at a single amino acid in a single mutation to the RAS to the RAS protein structure, right? So essentially, to like combine the information for like every single amino acid inside that structure, we had to generate a residual profile vector, which was like a very comprehensive vector that contained all the different residual scores at every single amino acid position. And so that was, we had a residual profile vector, which, so in total for the RAS protein structure, by the way, there's 166 amino acids. So the residual profile vector was a 166D vector, meaning it was like 166 dimensional, and it contained information about every single amino acid. But then we also generated a more localized vector. It was called a local profile vector, and that was only 27 dimensional. So that just had like more meaningful information, and it didn't include information about every single amino acid position, because that's also kind of logical, and it actually turned out to produce better results than the residual profile vector once we put them into the machine learning models. Because essentially, like with the residual profile vector, it's not necessarily beneficial to have too much information, because if you are including amino acids that are like way far away from like the original amino acid that was mutated, then like obviously there's not going to be that much of an effect on that like distant amino acid. So it was actually beneficial to just remove the information completely because it was actually messing up the machine learning models. And then on the bottom left, you can see kind of a visualization of of one of the residual profile vectors. So this says for mutation Q99Y, which means at amino acid position 99 out of 166 in this RAS, in this native protein structure of RAS, it switched the original amino acid, which was a Q, to a new amino acid, which was a Y. Okay, so in the previous slide, I was talking about the residual scores and the residual profile vector, but I didn't really go into detail how we get the fundamental block for the residual score, which was calculating the energy score. So basically, to calculate the energy score, we relied on a previously generated 4-body statistical potential for that other researchers had created in the past. So the way they created this was they essentially selected over 1,400 3D protein structures from the online protein data bank. And with each of these structures, first of all, they're selected specifically and particularly to have certain features. So ultimately, the 4-body statistical potential could be as informative as possible and as accurate as possible. So once they selected all these 1,400 plus 3D protein structures, they essentially applied a biophysical principle known as the inverted Boltzmann principle. And what this did was it actually provided the mathematical energy score for each of those quadruplets of amino acids inside of a mutant. So the actual formula is listed below in that diagram. So I'm not going to go into detail about exactly how it works, but just some basic information. That's a multinomial probability distribution function. So we had to use, or the researchers, when they created this potential, they used a multinomial distribution instead of a binomial distribution because the amino acids, they come in quadruplets, right? In pairs, in sets of 4. So a binomial distribution function wouldn't have accomplished that same thing. And then to create the actual, and that P sub ijkl, that essentially is equal to the expected and theoretical frequency of that particular quadruplet of amino acids occurring at random, but that's just theoretical, right? So that's why we have to use that distribution function. But then for the first metric, or for the first number, f sub ijkl, that is essentially the observed frequency of the quadruplet of amino acids. So this is in the real protein, in the real structures that they've obtained, how many times do they actually see that particular quadruplet of amino acids? So then ultimately, the energy score for that particular quadruplet was calculated by taking the logarithm of the observed frequency over the expected frequency. And that provided the energy score, which was basically the basis for the computational mutagenesis technique. Okay, so that basically sums up the computational mutagenesis technique. So after in this research, after we performed those tasks, the next thing to do was to actually validate and make sure that our technique was working properly. So one way we would be able to do this was through ensuring that the computational mutagenesis methodology captured the structure function relationship that's inherent in every single protein. So if it was able to capture this information, essentially we know that we're on the right track. So to do that, we essentially created this graph you see right here, and that just shows the fitness. The x-axis is essentially a measure of the structure, and the mean residual score on the y-axis is essentially a measure of the functionality of the resulting mutant. So essentially these bar graphs are in line with what we wanted. And again, I'm not going to go into too much detail here, but we split up the 3135 mutants into conservative mutations and non-conservative mutations. So the conservative mutations are denoted by C and non-conservative by NC. So essentially a conservative mutation is when there's an amino acid mutation in which the original amino acid is mutated to another amino acid that's in the same biochemical property of groups of it. So polar groups of amino acids, for example, if there's a mutation within that group, then that would be classified as a conservative mutation. Otherwise, if it's from like one group to a different group, then it would be non-conservative. But yeah, that's just the structure function relationship. And this is also just a different representation of the structure function relationship. It's just, again, the residue environment score is a representation of the structure of the protein, and the residue CMP score is, it denotes the functionality of the resulting mutant. And this diagram is also able to split all those different mutations up into the 3 different residue types. And you can see that the R squared value is quite high. It's 0.83. So that indicates a pretty strong relationship, meaning that, like, again, like our computational mutagenesis technique worked pretty well. Okay, so that's basically the end of the computational mutagenesis technique. And now here's the machine learning part. So this is just a flow chart of like the different ML models we used in the research. So basically, like the models, they're of course divided into 2 types of models. One is classification, and number 2 is regression. So basically, for classification models, we utilized random forest classification, and then for regression models, we used rep tree regression, which is a pretty like unknown regression model technique, but it actually worked pretty well for our research. So the 16060 and 2070, they just represent the different vectors that we inputted into the ML models. So 16060 is, as I said before, the residual profile vector, and the 2070 is the local profile vector. So we essentially like inputted both of the vectors in for classification models and for regression models, so we could see like just every possible combination, and then ultimately select the ML model that predicted like malignant versus benign the best. Okay, and then here's a summary of the ML models. So basically, the 6 metrics you see on the 6 headers you see in the top, they each represent like an ML model performance metric. So just to like measure how well the ML model did. And it's like not just simply like number correct over total number of cases, because that can sometimes not be optimal. Because, for example, if you have like a really, really bad ML model that just says like malignant for every single case, but then like the data set is like 90% malignant, even though that ML model is essentially guessing at random what the functionality is going to be, it would still get like a 90% based on that like that metric of just like saying the number correct over the total number. So for that reason, like because of like the potential discrepancies that could be inside of a data set, we have to use these more advanced metrics. So for the classification models, the best metric you would want to look at is probably going to be the one that's called AUC. That basically means area under the ROC curve. And you can't use an AUC for a regression model. So for regression models, we use the BAR and the MCC metrics. So BAR is the balanced accuracy rate, and the MCC is the Matthews correlation coefficient. So for classification models, you can see that like the first one worked the best, the residual profiles one that had random forest classification. And then for tree regression, it was residual profiles again. Okay, and then here's a summary of like the final output of our research. So this is essentially like a data plot with every single one of the 3,137 mutations to the RAS protein structure. And it essentially just shows like a clinical doctor could essentially just like point to this, like trace the mutation and see whether it's malignant or benign. So it's just like a very organized way to see like whether the mutation is bad or not bad. And then here's just another table that goes over like a comparison between our models and the other online models we found on the internet. So these are just like other state-of-the-art methods that we compared our thing to. and we're actually able to outperform all of them. Okay, so now the next few slides are just going to be a bit of data analysis. So in figure A, that's the area under the curve I was talking about before. So essentially, a line that just goes straight through the diagonal indicates a model that just randomly guessed the functionality of the resulting mutant. So that's pretty bad. You don't want that. But then like the optimal ROC curve is one that kind of goes to the top left, which you can see is our thing. It has that nice curvature. Okay, then here in figure B, this is basically a graph that establishes the statistical significance of our models and like consistently outperforming the random model over 1,000 iterations. So it just shows like our two bell curves and just shows how like we are like over 1,000 iterations, we're able to consistently beat like a model that just guesses at random. So it kind of just like proves that like our models did do something, right? Okay, then here's figure C, which is just a scatter plot that reports the results of a particular ML model. So in this case, it was the regression model that used rep tree regression, and it used it on the local profiles vector. So basically, if every one of those dots represents a different mutation, so in total, there's over 3,000 dots in this diagram. And essentially, if a dot lied in the first or the third quadrant, that means we correctly predicted it as either being malignant or benign. And if it lies in the second or the fourth quadrant, then we incorrectly predicted that mutation. So you can see that the density of dots is much greater in the first and third quadrants. And yeah, that's just a good sign for our model. And you can also see that the correlation coefficient is 0.79, which is pretty good for a model. Okay, and then one final data analysis graph that we created was a learning curve. So this isn't really that important, but it does like provide useful insights for like future research. So what a learning curve shows is basically like how the accuracy of your models improves as you add more data points to your training set. So essentially like what the difference in accuracy of the ML model was when it had 1,000 data points versus when it had 500 data points, right? So in this case, like it tells us that it doesn't really matter if we had 3,000 versus 3,500 data points, because you can kind of see that the curve just kind of flattens out eventually. So at that point, the accuracy didn't really change that much. It wouldn't really make a big difference if we had more data points. Okay, so in conclusion, like we showed in table 2, the models were able to outperform all those different state-of-the-art methods, and it was able to account for the downsides of the previous research by providing a simple, inexpensive, quick, and efficient technique that also didn't use plastic for predicting the functional effects of RAS mutants. It also is like a proof of principle study for future research, just into understanding more how exactly these RAS mutations work and how they like eventually lead to cancer. 3 clinical applications. So yeah, this could be really helpful for doctors. It provides really useful insights into targeted therapy and like before the clinical trials can even begin. And then in terms of future work on this project, there's a few things that we could have done that we didn't do. Number one is investigating multiple point mutations in RAS. As I noted before, we only looked into these single point amino acid mutations, but like double, triple, and multiple point mutations do exist, and they do lead to cancer. But at the same time, it probably wouldn't make a huge effect just because like single point amino acid mutations make up by far the most number of mutations in RAS. We also could have looked into like other proto-oncogenes. So basically, like RAS isn't the only protein that eventually causes cancer. So we can essentially like replicate this research methodology when we're analyzing like other proteins and like get more insights into how cancer develops. We could do targeted therapy, which is like directly collaborating with pharmaceutical researchers to actually develop those inhibitors that would turn off a mutant that's like in a permanently on state. And of course, that would be like solving cancer at its root, because like automatically prevents tumor growth and cancer development. And then number 4 is clinical data. So working with clinicians to gather patient genomic data to further refine our ML models that we have. But I'm not sure if that's like entirely possible, just because like some patients might not be comfortable like being the subject of like different research. Okay, so that was like definitely the most complicated application we're going to go into today. The second application is going to be on predicting gene expression from nucleotide sequence. So as some background, DNA it makes up the genetic code of all living life. It's essentially first transcribed into mRNA and then later translated through the codons into amino acids, which then form the proteins. And then the proteins are able to produce the phenotypes of an organism. And the phenotypes are just kind of like physical characteristics like eye color, hair color, things like that. So gene expression, like the final result, is affected by several factors besides just the DNA nucleotide sequence. But looking at the nucleotide sequence usually provides like a pretty good prediction for what eventually the phenotype will be. But at the same time, like current techniques, they're kind of limited by the fact that they can only look at the nucleotide sequence and not look at other factors. So the problem is that gene expression profiling is currently used in like a wide variety of settings, but the problem is that like it's pretty high in cost with the current laboratory techniques. So it's pretty crucial to have cheaper and more scalable options. And of course, like the best way to do that is just to switch to like computational techniques. So here are 2 potential like algorithms that could be used to accomplish the same task and possibly even produce better results in predictions. Number 1 is the XGBoost algorithm. So this is just like a variation of like a basic machine learning decision tree model, but it has certain optimizations that are like pretty helpful. Number 2 is KNN, which is it's not an ML model, but it's just like a computational technique that uses the formula you can see down below. So in terms of further applications, through using genetics and studying the sequence of other cancers, oncologists can possibly predict new cancers from just the nucleotide sequence of DNA. Mutations in the human genome can be compared to the completed sequence genome to find any changes in the original gene expression. Okay, and then understanding functions of sequences. So like even though the entire human genome has already been mapped out, there's still like certain sequences that researchers don't completely understand. So essentially predicting like what effects, like what final gene expressions those sequences have, it can just help in predicting different phenotypes and like understanding what exactly those sequences are responsible for. Okay, and then the third application is protein folding. So as some background, sorry, 1 second. Okay, so as some background, yeah, proteins are just amino acid chains that fold up into different structures. There's several structures of a protein: primary, secondary, tertiary, and even quaternary. So primary structure is just like the DNA sequence, basically. And I'm gonna have some diagrams on like a few slides from now that will kind of illustrate this. And basically, protein misfolding is when the wrong amino acid is inserted into a chain, and essentially, like when this happens, protein misfolding, like diseases like Alzheimer's, can be potentially caused. So bioinformatics can be key in helping us predict what the larger structures of proteins can be based on like a multitude of factors instead of just like single factors. And yeah, this is actually pretty key because like currently like computational techniques can only look at, or sorry, not computational techniques, like more laboratory techniques can only look at like sequences of up to 100 DNA nucleotides. So they're kind of limited in like how far they can go. And that's kind of why you need computational techniques. Okay, so here's an illustration of like the levels of protein organization. So first you have the primary one, which is just like the sequence of amino acids. Then it's secondary, which is like the alpha helices and the beta pleated sheets, and then it goes into tertiary and quaternary. Okay, so the problem is that proteins, in order to find their final structure, they have to search through like an extremely large number of conformations. So there's like kind of a twofold problem here for researchers. Number one, like what exactly is the precise mechanism for which like they find the final conformation? Number two, is it possible to predict the final structure, like the quaternary structure of a protein, from the original primary sequence? And if so, how? So for the protein folding code and mechanism, there's like, it's a pretty complex issue because there's multiple factors going on. Number one, like environmental factors and like pH and temperature, chaperone proteins, and like hydrophobic interactions. Okay, so here's one computational technique that can be used. Basically, number one is a PSV plot. So this kind of relies on the fact that the folding speed of a protein is directly correlated with the protein structure. As you can kind of see with the scatter plot on the right, has a pretty high R value. So what this means is that like fast folders tend to have local structures like alpha helices, and slow folders tend to have non-local structures like the beta pleated sheets. So that's number one. And then number two is funnel shaped energy landscapes. So these aren't actually computational techniques. This is more of like a physics technique, but it does like the initial data processing has to be done on a software like MATLAB. So it is somewhat like a computational biology application. Yeah, so yeah, other than computational techniques, physics-based modeling is often used in these computer simulations. So applying like different physics concepts like REMD to sample conformations. And as I stated before, like currently the models and the technique, like the lab techniques, they're only effective for proteins with chains up to 100 monomers. So more research is required to kind of see like what the structure prediction would be for larger proteins. And there are a lot of applications of being able to predict protein folding correctly, because like I said before, Alzheimer's can potentially occur when a protein is folded incorrectly. So like predicting these diseases ahead of time can obviously help doctors prevent these diseases from occurring in the first place. Yeah, and also just provides more insight into understanding exactly how proteins become denatured, because this is actually a big thing that's not really been found out in biology. Okay, and the fourth and final application I'm going to go over today is going to be brain biometrics. So basically the current problem is that like data breaches are very prevalent. So around according to a Pew Research Center survey conducted in 2016, 64% of US adults have experienced a breach in their lifetime. And of course, like these breaches, they can have detrimental effects. So like one research group found that the annual loss due to cybercrime is actually $445 billion per year. So basically, in order to prevent these data breaches, people have started to use biometrics. And if you don't already know what they are, they're just basically like physical traits that are unique to every single person. So this could be like your fingerprint, your face, your iris, even like voice recognition system. But while these have been like somewhat effective in the past decade, they're also like there's certain weaknesses that are kind of starting to be like exploited inside of them. So number one, traits are immutable. So what this means is that like if someone just like is able to take your fingerprint, like not actually like cut your thumb off, but like, you know, like, I don't know, like put it on some kind of paper and like just, they have a copy of your fingerprint, right? Then basically they like, you can't really use your fingerprint as a biometric for the rest of your life because someone else has it, right? The same thing can go with like iris. I don't know, if someone has like a picture of your eye, or even voice recognition systems and facial recognition systems, the same thing can happen. So yeah, they're compromised forever after like just one hacking. Number two is that these traits that are used are public. So there's actually an interesting thing that happened a few years ago. A German defense minister through high quality photographs, she had her fingerprints reverse engineered just from photographs. So again, that's just like is somewhat related to the immutability, because like once those hackers obtained those photographs, she couldn't ever use her fingerprints as like a biometric again, right? And then number 3, these traits are exploitable. So like someone could like physically pressure someone into authenticating into a system. So yeah, that's definitely a huge issue. Okay, so one area that's not really been looked at is in biometrics is utilizing the brain as a biometric tool. So the way this works is kind of dependent on like a few properties of like every human's brain. So basically, whenever the brain is like whenever someone's exposed to a stimulus, the brain response that's occurring is always going to be unique and consistent. So it's going to be unique in that like no other person can have that exact response that you had, and then it's going to be consistent because like if you're showing that same stimulus multiple times, your brain response is going to be about the same every single time. So because of those two properties, it's like an ideal candidate for biometrics use. So in order for it to actually work, it requires the use of an electroencephalography test, an EEG test. And in one implementation of brain biometrics, what could happen is that the user's auditory evoked potentials are measured in their brain, and that essentially creates a unique brain signature for every user. So then like when the user is trying to re-authenticate into the system, essentially their AEP would be measured again. And if it matches up with the original AEP, then the system knows that it's the real user that's trying to enter the system. So the unique value of brain biometrics is like it kind of addresses the 3 major problems that were outlined before. For some like public traits, this is obviously private because like you can't take a photograph of someone and like suddenly see their brain because it's inside of them, unlike like fingerprints and iris and stuff. Number 2, it can be changed. So in contrast to the immutability of certain traits, for brain biometrics, you can actually change your authentication pattern by just exposing the user to a different stimulus, because then you get an entirely new response that's unique to you. And then number 3, on the third one is exploitability. So on exploitability with EEG data, something that's actually pretty interesting is that like when you're under stress, like under certain emotions, your brainwaves are actually like altered. So you wouldn't be able to re-authenticate into the system under that current state. So if someone's like physically pressuring you to authenticate into a system, essentially like you'd be under a lot of stress that you would not be able to be validated into the system. So it protects your data. And then the solution for brain biometrics is basically, it's a 4-step process. So first, you have the initial EEG test with acoustical stimuli. And then from that, your brain signature is registered. And basically, when you're trying to re-authenticate, you just use the same stimulus and you measure the same brain pattern. And then you're able to, like, behind the scenes, there's some like deep neural networks that's going on, and they're going to either grant or deny you access to the system. So there's like a lot of impacts of this, of brain biometrics. So first of all, in terms of its market, companies with highly private user data could definitely use this, like banks, hospitals, and stuff. Government agencies with highly classified data, of course, this would also be beneficial to them. And the thing is also that, like one downside right now is that the test for an EEG is probably going to take like 2 minutes. So you're probably not going to see like brain biometrics being applied to your phone, for example, like to unlock your phone or your computer. But definitely for right now, like it could be a huge help in terms of like companies and government agencies that have highly classified data. And then another impact is, or another like benefit of brain biometrics is that it has the potential to achieve 100% accuracy because of how unique and consistent every person's brainwaves can be. So this 100% accuracy has not actually been achieved yet, but it does have the potential for it. And like currently other forms of biometrics, like fingerprints, for example, they actually cannot, they do not have the potential to reach 100% accuracy. And then in terms of cost effectiveness, this is also like relatively cheap, because like high-end fingerprint scanners that are used for like highly classified data, they can be like $20,000. But then for brain biometrics, the entire package can come in like $400 to $500. Yeah, but basically, those 4 applications kind of just illustrate like the wide range of scenarios in which computational biology can be applied. So like initially we were looking at like a very specific research topic, like computational biology being applied to predict the functionality of mutations to RAS proteins. But then now here, like in the last application, we can kind of see like a very real world application of computational biology in brain biometrics, which like has a really big market and can affect like a lot of people. But yeah, I think computational biology is like a rapidly growing and really exciting field, and it's definitely like a good time to get into it. And like a lot of you, even if you have like a slight interest in bio or CS, I would definitely recommend trying to like look into it, because I think a lot of you might be interested in it. But that's about all I have for the workshop. So thank you for listening, and I can take any questions that you have right now.
All right, thank you so much for that wonderful workshop. We do have one question. For EEG, how long do you believe it would take for authentication to take place? A person's mental state changes from day to day, and as people can have developed psychological illnesses, do you think that they could easily mess up the authentication process?
Okay, so there have been like a few papers in like South Korea that have kind of explored this and made like preliminary brain biometrics testing, and basically they were able to do it in around 2 to 3 minutes. But yeah, definitely 2 to 3 minutes is still fairly long, and that's why you're not going to see the applications and like unlocking your phone or computer. But definitely in terms of like the really highly classified data, it could still be a huge help, even if it's like a few minutes long. And yeah, I agree. Definitely like a person's mental state changes from day to day. But at the same time, like I was exaggerating a bit when I meant like if you're just under a bit of stress, you're still going to be able to get authenticated into the system, because like still every person's brainwaves are just so distinct from like another person's that the computer will be able to tell the difference. But yeah, like in terms of like highly stressful situations, that would kind of mess up the authentication process.